In the second part of my presentation, I will talk about the structure of the program more detail, especially the use of the structure of the software for association mapping of the bacterial spot resistance in tomato. Bacterial spot is a major disease in tomato. It is the disease complex caused by the several Pseudomonas species. 5 physiological rates have been identified for this disease. There are several tomato lines showing the race-specific resistance, but we need non-race-specific and durable resistance in the elite the background. So we initiate a research for simultaneous the mapping and the pyramiding of the bacterial spot resistance in breeding population. In this study, we employed association mapping approach for marker and trait association analysis. For marker and trait association analysis in breeding population, you should consider population structure, otherwise you may detect false positive association. This correction can be done by adding the Q matrix from structure analysis in association analysis model shown here. So in next several slides, I will go through the steps how to generate the Q matrix using the structure. First step of the structure analysis is the format of your data. Here is the input data in structure format. In the first part of my presentation, I explained how to format the data for the structure, so I will skip it here. And this data is available at the workshop, the URL in the PBPG web. The second step is to decide how long to run the structure by setting Bonin rank, the Bonin and MCIC ranks. Bonin ranks is how long to run simulation before collecting the data to minimize the effect of starting configuration. Recommended the Bonin ranks is 10,000 to 100,000. MCIC ranks is how long to run the simulation after Bonin to get accurate parameter estimate. Recommendation for these ranks is the 500,000 to 1 million. After setting Bonin and MCIC ranks, run simulation 20 times for each of the several different K to identify the best K. The K here means number of population. Based on the log likelihood value from the 20 simulation, you can identify the best K using the non-parametric method, or the delta K method. The logic behind the non-parametric method is when the K is approaching a true value, the likelihood value plateau or continue increasing slightly and have the high variance between log. In this top graph, the likelihood value increased dramatically from the K equal 1 to 5. But after 5, there are very little change, and you can see nearly the huge variation between lots. Okay. The non-parametric test actually find where is the first place the mean difference between successive likelihood value is not significant. The second approach to identify the best case is to use the ad hoc quantity, the delta K. In this method, when you plot the delta K, the value, you will see clear peak at best case, shown in this bottom of the graph. This is a screenshot showing the simulation summary from the structure analysis. In this simulation summary, you can find the log the likelihood value in the first column. And once you find the log the likelihood value, then bring them in Excel spreadsheet to calculate the delta K, the delta K. Here's the example of the calculation for the delta K. The first, calculate the average of the 20 the likelihood value. Then calculate the prime the LK, which is the mean difference between successive the likelihood of the value of the K. Then calculate the double prime the LK between the prime the LK. Finally, you can calculate the delta K by dividing the absolute value of the double prime the LK by the standard deviation of the likelihood value. In this graph, you can see the clear peak at the 8. Actually, we calculate the delta K for the 12 difference K. Then we plot here. And we observe the clear peak at the K equal 8. It means our predicted population has the 8 subpopulation. This worksheet is available at the SourceCap, the workshop URL. The last step is to retrieve Q matrix for the best case. This is another screenshot showing the screenshot of the structure, the program. At the left window, you can see the simulation you did. From here, find one of the 20 simulation for the best case. Then at the right window, you can see the detailed output of your simulation. And also this is the bar plot showing the graphical representation of your Q matrix. And if you scroll down, the output at the right window, you can find the inferred ancestry of the individual. That is a Q matrix you need. With that Q matrix, you can correct the population structure by adding the Q matrix in your association analysis model. Here, this is the code we use, and in the model statement here, we add the Q matrix to estimate the the more accurately the marker effect. This test code is also available at SourceCap workshop URL. This slide summarizes the second part. The structure is a useful tool to detect the population subdivision. The use of the Q-matrix can correct for subpopulation during association analysis in breeding population and avoid the detection of the false positives. The SNP results from the SourceCap are a powerful survey tool, so we should be thinking beyond the biparental population toward the analysis of the complex breeding population. That is all that I have, so I'll be happy to take any questions.